Development of neural tube. Neurulation, process of formation of neural tube, occurs in the third week of life after gastrulation. Formation of three germ layers gives origin to brain and spinal cord. Formation of neural tube. Ectoderm, mesoderm, endoderm. Notochord gives signals to ectoderm. Ectoderm thickening to form neural plate. Neural plate invaginates due to signals from notochord. Neural groove formation. Neural crest cells at the junction surface of ectoderm and neuroectoderm. Surface ectoderm not involved in neural plate formation. Ends of neural groove fuse to form neural tube. Neural tube is formed earliest at the cervical region. Sclerotome surrounds the neural tube and notochord, gives origin to axial skeleton. Neural crest cells and mesoderm migrate to different parts of the body. Derivatives of neural crest cells. Sclerotome, paraxial mesoderm form somites. Sclerotome, myotome, dermatome. Neural tube, neural crest cells, fourth germ layer. Fourth germ layer is coming from here. So the ectoderm starts thickening, causes the neural plate which invaginates and forms the neural tube. This at the junction of ectoderm and neuroectoderm is neural crest cells which migrates to different part of the body and gives derivatives of the neural crest cells. Neuropores communicates neural tube with amniotic cavity. Cranial neuropore failure to close, anencephaly. Caudal neuropore closes by 28th day. Failure to close, spina bifida. In between neural tube defects due to deficiency, these are known as neural tube defects due to deficiency of folic acid, prevented by giving folic acid to pregnant women in first trimester. Dura mater, spinal cord, spinal fluid, vertebra. Dura mater, spinal cord, spinal fluid, and vertebra. Cranioarchesis, severe form of neural tube defect. Entire neural tube is open, that is cranioarchesis. Neural tube derivatives. Cranial part forms brain. Caudal part forms spinal cord. Three primary brain vesicles. Prosencephalon, forebrain. Mesencephalon, midbrain. Rhombencephalon, hindbrain. Prosencephalon again, telencephalon and diencephalon. Mesencephalon gives mesencephalon. Rhombencephalon gives metencephalon and myelencephalon. Lateral view of neural tube. Prosencephalon, mesencephalon, rhombencephalon. Forebrain, midbrain, hindbrain. 3 to 4 weeks embryo. 5 secondary brain vesicles. So from the prosencephalon, you have again the telencephalon here, which will form the cerebrum. You have the eye cup from the diencephalon. Diencephalon also give rise to thalamus, hypothalamus, and epithalamus. Then in the mesencephalon will give rise to midbrain, and the rhombencephalon, you have metencephalon, will give rise to pons and cerebellum, and myencephalon will give rise to medulla oblongata. here. They have given a table. So primary brain vesicle, prosen, meson, and rhomben. Prosen gives talon and dian, secondary brain vesicle. Derivative, talon, cerebrum, corpus striatum, dian, thalamus, hypothalamus, metathalamus, subthalamus, epithalamus, pars nervosa, posterior lobe of pituitary. Optic stalk forms retina and optic nerve. Cavity, telencephalon, lateral ventricle, big ventricles, and diencephalon, third ventricle. Mesencephalon, midbrain, aqueduct of Sylvius. Rhombencephalon, metencephalon, and myelencephalon. Metencephalon, pons and cerebellum. Myencephalon is medulla, fourth ventricle.